the recent years, we have come to understand that individuals without hematologic malignancies accumulate mutations in their blood stem cells, even though by the various clinical parameters, the individuals do not display any abnormalities in their blood production or hematopoiesis. We have come to know this as clonal hematopoiesis. Importantly, the mutations that we observe in clonal hematopoiesis are the same ones observed in acute myeloid leukemia. This means that we have potentially the ability to examine the phenotypic impact of cancer-driving mutations at the earliest stages of the neoplastic process and prime the affected cells for malignant transformation. Various labs have shed light on the significance of these mutations using beautiful animal models that have provided us with really profound understanding of what these mutations are doing in the blood cells. However, the direct examination of these mutations in human clonal hematopoiesis has been challenged by the fact that the mutated cells are basically indistinct by current modalities such as FACS from the wild-type cells that are mixed in these individuals. So to overcome this challenge, we leverage the technology that we developed recently in which we can capture the mutation status as well as the transcriptional states of the thousands of cells from these primary human clonal hematopoiesis samples. And we focused on a gene called DNMT3A, which is the most commonly mutated gene in clonal hematopoiesis, and specifically a mutation R882, which is a hotspot mutation strongly associated with acute myeloid leukemia. So by applying our single cell technology that we termed genotyping of transcriptomes, or GOT, we could compare the transcriptional states of mutated and wild-type cells within the same individual. And the advantage with that is that then we have a perfect comparator for these mutated cells, such that we overcome all biological and technical batch effects that would prevent us from observing the truly biological signals that we want to extract from the mutated cells. So by applying this technology, we were able to understand what are the sort of the perturbations to the differentiation topologies caused by the somatic mutations, specifically the DNMT3AR882 mutation. And we identify that the DNMT3A mutation causes differentiation skews at key junctures of hematopoiesis. For example, the DNMT3A mutations cause a myeloid versus lymphoid bias at that sort of decision point, as well as that the mutation seems to bias these early myeloid immature progenitors toward the megakaryocytic erythroid lineage fate. Furthermore, we could compare the transcriptional states of the mutated and wild-type cells and identify what are genes that are upregulated by the mutation, of the gene expressions that are perturbed in these mutated cells. And we identified actually markers that were identified to be upregulated in acute myeloid leukemia stem cells, such as CD99. And we were particularly excited about this finding because it's a cell surface marker that could that actually has been proposed as a therapeutic target in AML. And this finding suggests that we may even be able to target clonal hematopoiesis stem cells through markers such as CD99. We further applied another technology in which we could capture the mutation status, the transcriptional states, as well as the methylation states of these individual cells from patients with clonal hematopoiesis, so that we could understand how does the mutation in this DNMT3A, which is a methyltransferase, affect the transcriptional perturbations that we observed. We identified specific regions that the DNMT3A mutation particularly impacts, such as enhancers and CPG islands. we identified that particular TFs actually may be more prone to be dysregulated due to hypomethylation of its binding motifs, such as MYC. We also actually, which is significant because we saw MYC sort of aberrant activity within our transcriptional data as well. So altogether, these technologies enabled us to examine the impact of DNMT3A cancer-causing mutations in the earliest stages of the neoplastic process within the primary human samples.